In this video, we will discuss about this therapeutic efficacy on genetically engineered mice. In the previous video, we have understood about this pharmacological efficiencies of combination therapy, and in that case, what we have done is we took the tumors from the genetically engineered mice and do the orthotopic injection into the animals or in the another healthy mice, which is the immunodeficient mice. So however, the problem in that approach is that the tumors that developed in those mice are not reflects the actual tumor progression in the humans, whereas the genetically engineered mice are more close to reflects the disease the way it progresses into the humans. That's why they wanted to do this therapeutic efficacies directly on the genetically engineered mice. For those who have not gone through our previous videos, the genetically engineered mice is the mice that in which they have knocked down or they have used the cleaved based knockdown or inhibition of two genes, that is RF1 and EGFR, and at the same time they can also knock down this another gene which is important, that is STAT3 using shRNA. So they use that approach in the initial studies, especially during the in vitro studies, to check whether this gene knockdown will prevent the tumor cells resistance against the cell death or not. So they genetically engineered those genes into the mice, and then what they did in this approach is that they allow the tumors in the genetically engineered mice to reach up to 100 mm to 300 mm, and thereafter they started treating these mice using different therapies. So in this case, they have used two different approaches. First, they used the monotherapy using this darax, and in the combination therapy they used all the three drugs, that is darax, AFA and SD36. The full name are in the descriptions. You can refer to them to get the full name of these drugs. So in the darax, I think there is some typing mistake. It should usually they call it as darax in the short form. So in this case, they have seen the similar results that they have seen in the pharmacological study where they used monotherapy using darax. That when they use darax, there is a delayed tumor growth or tumor suppression is there, but the tumor cells is not able to tumor cells still have this has the ability for resistance against the cell death. So in that case, what happens, although the tumor suppression rates are low, but eventually those mice died because of the tumor ability to resist cell death. However, when they used all the three drugs together, or the term that they use is combination therapies, where all the three drugs are used together, they are able to see tumor suppressions, and they checked it till day 200, and they can see that around half of the tumors that included in this half of the mice that included in this experiment, that is around 12 mice, do not show tumor relapse even after 80 to 200 days. And tumor relapse results for two different time point are shown in this panel B, where we can see that at day 20 the tumor volumes are higher in the case of vehicle, which is a control which doesn't undergo any treatment, and also in the monotherapy which sorry, and also in monotherapy where this mice undergoes only this darax. There also the tumor volumes are higher than at the point when they have started using the drug. Whereas in the case of this combination therapy, they are able to see tumor suppressions almost in all cases, and by day 80 they are able to see that tumor suppressions are still continued even after 80 days, and this empty vector represents the tumors is completely eliminated within some mice. So, and when they check the reason for this tumor suppression, and from the previous results, they have an idea that tumor suppressions usually occur. Tumor suppressions usually reduced because tumor cells undergo apoptotic or programmed cell death using apoptosis. So in this case also they checked the apoptotic marker, it is cleaved caspase 3, and they are able to see that in the combination therapy the markers for the cleaved caspase, the signals for the cleaved caspase 3 are high compared to the vehicle, indicating that these tumor suppressions are because tumors lose their ability for resistance against the cell death, because all the 3 genes that plays a role towards resistance are being suppressed. So this is what they conclude from these results. So the final conclusions that they find out that therapeutic efficacy is as much effective as they see this combination therapies. Therapeutic efficacies of combination therapy is as much effective in the genetically engineered mouse as they have seen in the case of pharmacological study. So far their results are seems promising and probable, and for this combination therapy to be used for the clinical trial. However, we will discuss further results in our next video to see what are the additional tests that they did to confirm the targeted killing of the tumor cells or targeted therapeutic efficacies against the tumors. So I hope you understood to some extent how they did their therapeutic efficacies validations. And if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.